Hello, everyone. We are once again in the South American Mycorrhizal Research Network YouTube channel, which is called What We Are Reading. And this time around, we have a very interesting group of people talking about hyphospheres. We have Professor Gu Feng and Professor Nancy Collins Johnson. So maybe can you both of you introduce yourself very briefly?
Yep. Yep.
I'm Nancy Johnson, and I'm a professor of ecology at Northern Arizona University.
And
Okay.
you?
Hello. I'm Gu Feng. I'm from China Agricultural University. I'm a professor of soil science and plant nutrition. Yeah.
Great. So today we are talking about hyphospheres and specifically about two recent really interesting papers by group. Um, One is entitled "A Core Microbiome in the Hyphosphere of Arbuscular Mycorrhizal Fungi Has Functional Significance in Organic Phosphorus Mineralization." And the title of the second one, which is a translate inside in both of them are in Phytologist, is entitled Concepts and Consequences of the Hyphosphere Core Microbiome for Arbuscular Mycorrhizal Fungal Fitness and Function. So the link of both of the articles will be in the description of the video. And with that, I'll start with the first question. So, Gu, how this paper came about and under which context, project, or under which set of ideas did you came with these two papers?
Okay. So the idea of core microbiome and in the hyposphere that comes from our previous studies of vascular mycorrhizal fungi and bacterial interactions. So when we look at the abundance of the phosphatase producing bacteria in the hyposphere microbial community, we often find some interesting things. For example, the most of the species, here I mean the OTUs or ASVs, so usually vary greatly among the different treatment. For example, the different soils, different P sources. Sometimes we supply organic or inorganic phosphate. So that is called P sources or EM fungal species. So we even find that different AM fungi, different AM fungal species recreated distinct microbiota community. So, however, we can also find some bacteria that always appear in different samples and different experiment. So in our previous studies, we always use different soil that's collected from different sites. So that's very interesting. So we find this phenomenon. So some bacteria taxon are often higher abundance, such as the streptomyces. So therefore, I guess these might be a group of bacteria species that might be commonly shared by different AM fungal species. So this inspired the idea of exploring the hyposphere core microbiome.
Could you elaborate on exactly what the hyphosphere is and define it for us?
Okay, so in our recent published National Review paper in Plant and Soil, we defined the hyphosphere as a critical and active zone of soil that's surrounding the living extra-radical mycelium of arbuscular mycorrhizal fungi beyond the rhizosphere and mycorrhizosphere. This zone is directly influenced by exudate of mycorrhizal fungi. So I would like to stress that the physical, chemical, and biological characteristics of the soil in the hyphosphere are different from bark soil due to the influence of heavy exudate. Yeah, that is the definition.
This is so interesting. And do you think this is exclusive to AMF, to arbuscular mycorrhizal fungi, or is possible that there are also hyphosphere for ectomycorrhizae or ericoid mycorrhizal fungi?
Okay, so I don't think the hyphosphere is exclusive for AM fungi only. The other type of mycorrhizal fungi, such as the ecto mycorrhizal fungi, can also form their hyphosphere zones. Actually, the ecto mycorrhizal fungi form even more extensive hypho networks in forest soils. I think their hyphosphere effect might be even more stronger. For non-mycorrhizal fungi, I think including the subtrophic and pathogen fungi, I think they can also release the different type of exudates like enzymes to decompose the soil organic materials and even soil root tissues. So these process can induce some changes of the soil characteristics and affect the soil structures and the nutrient availabilities also.
Sure.
So in my point of view, so we should limit the term of hyphosphere to mycorrhizal fungi induced changes of soil conditions. So we should not include the non-mycorrhizal fungi. That's my point of view. So unlike the non-mycorrhizal fungi, the mycorrhizal fungi can continuously get fresh carbon from living plant. So these induced change in hyphosphere are consequences of positive actions of mycorrhizal fungi and their host plant. So that's why I said so we should not include non-mycorrhizal fungi.
I see.
Yeah.
Could you elaborate a little bit more about what the ecological and biochemical processes are occurring in the hyphosphere?
Okay. Yeah. As we know, although the single mycorrhizal fungi hyphae is hard to be seen with our naked eye, the fine hyphae induce the ecological and the biological effect in hyphosphere are visible actually and cannot be ignored. So the process happening there can be physical, chemical, and biological, such as promoting the formation of soil aggregate and the soil aggregate and the aggregate water stabilization. The mineralization of soil organic phosphorus and soil nitrogen, organic nitrogen. The hyphosphere process also involved in soil carbon sequestration and nitrous oxide emission from soil. So recreating and enriching some functional bacteria groups. So there are many different, I think, ecological or even environmental process that can be happen in the hyphosphere. One of the most predominant process in mycorrhizal fungi is one of the most predominant process is the mycorrhizal fungi have to recreate phosphatase producing bacteria to mobilize soil organic phosphate, because the fungi do not have the ability.
Yeah.
So for me, as a soil science and plant nutrition person, the study of hyphosphere process is important because it is one of two nutrient uptake pathway of plant. So we normally call the root pathway and mycorrhizal pathway. So the better understanding of the ecological process in hyphosphere can produce some new knowledges and technologies for ecological intensive cropping management.
And speaking of technologies, can you describe us a little bit how to actually measure or assess hyphospheres in situ, and if it's so far only possible in croplands, or if would be possible in the future also in forests or grasslands?
In forest, okay. Yes, so to measure the effect of the hyphosphere, I think it's difficult because the single hyphae is not be observed by our naked eyes. So we have to develop some method that make that can be the effect can be visible. So first in our work, so research work. So firstly, we developed in-groove tubes to isolate AM fungal hyphae from soil. So this helps us to identify the bacteria on the surface of hyphae and compare them with bark soil. That means we have to create a sample that is influenced by the hyphae and compare with the bark soil. So we use this method. We established the on-site field trials located in the humid, semi-humid, and arid zones. So we use the different differential analysis and the network analysis to identify bacteria due specific to the hyphosphere. So finally, we conducted a pod experiment, use a single AM fungal isolate to further validate the existence of the core microbiome. I mean, so for the device, we use the two type of in-groove tubes. So it looks like such kind of PVC tubes. So we normally use the two type of mesh. So one mesh is 30 micrometers diameter. So we covered these two ends of this tube. And for another mesh, so the diameter is 0.5 micrometers. So we use these two types of mesh to cover this tube. And the 30 micrometer nano mesh is can prevent the root to stop root penetrate, but allow the mycorrhizal fungi penetrate into the tube. So with this method, so we can create it we call the hyphosphere soils. And the 0.45 micrometer nano mesh can stop both the root and the mycorrhizal hyphae access into the tube. Then with this method we can create a non-mycorrhizal, non-hyphosphere soil, that in another term is box soil. So with this method, these two tubes, so we can get two types of soils, so the hyphosphere soils and the box soils. So then we can compare the effect of the mycorrhizal fungi that induced. That is the details of this method. Meanwhile, if we want to test the hyphosphere microbiome, so the mycorrhizal bacteria community that's living along the hyphae, so we have to get enough hypho materials. But we know that it's very difficult to isolate, to extract the single hyphae from soil. That is not possible, actually, in field. So we also use this method. So we put some piece of some piece of nano mesh in this PVC tube. So the nano mesh diameter is also 0.5. So the diameter is a little bit smaller than this tube. Then we put the nano mesh into the tube, and when the hyphae get into this ingress tube, so they will meet the hyphae, the nano mesh. They will be stopped there and grow cumulatively. Then they will have to turn around to go to another space of the soil and then meet another nano mesh. Then they will grow on the mesh and accumulate it on the mesh. So by this way, we can easily to collect, or we can say trap, a lot of mycorrhizal hyphae from that each piece of nano mesh. Then that will be very easier to collect them in lab. But one problem is so we cannot avoid the saprotrophic fungi to access into the ingress tube. And these fungi hyphae, these saprotrophic fungal hyphae can also enrich on the block membrane. So in order to reduce the influence from the saprotrophic fungi, so we have no good method currently. We picked up the saprotrophic hyphae, saprotrophic fungal hyphae under microscope by faucet. That is a very hard work, the time-consuming work, but we have no good method. So by this way, so it's very, I think it's relatively easier to identify, to separate the mycorrhizal, arbuscular mycorrhizal fungi hyphae and the saprotrophic mycorrhizal hyphae. Finally, we measure, we get purified the hyphae samples. So before we make a sequence, so we check the DNA, the ratio of DNA of AM fungi and the saprotrophic fungi. So our results show that the purified sample is acceptable. So by this way, yeah, we can get around 90% of purified AM fungi.
Oh, that's quite a lot. That's amazing indeed, and it's very hard and detailed work. Just to start in wrapping up, and maybe you already answered this in the first question, but and I invite our listeners to read the translate inside because they go really into detail. But can you tell us what mechanisms can maintain this core microbiome? Why there is a core microbiome in the first place?
Well, okay, what the mechanism to maintain this? Actually, I have no idea to explain. So how the this microbiome is maintained, in particular the core microbiome is maintained on the hyphae. I think this is still an open question. So we need to do in future. But I think so the interactions between this between this bacteria, so the is one possible mechanisms. Another mechanisms might be the demand of the fungi. For example, the mycorrhizal fungi cannot produce phosphatase. They don't have a phytase gene in their genome, so they have to recreate the phosphatase producing bacteria. So that is my possible mechanism that the fungi have to release some signal or some activate to collect the bacteria that the fungi really need.
Nancy?
So what are the next steps that you're taking in this very important area of research? And also for you personally, what are you doing next?
Okay. Right. So I think the hyphosphere is the concept of the hyphosphere is not new concept. It has been created many years ago, but recent years, so we have found some interesting things in this small tiny zone. So, but I think yes, there are still have many questions that we need to consider to do in future. Firstly, I think some questions I think is very interesting for me. So firstly, so is why and how the hyphosphere core microbiome is formed. For me, I still keep thinking about one thing I'm keeping about thinking about is so the bacteria and the fungi need each other. Yeah. So the fungi lack of phosphatase gene, that they need the bacteria to help them. I think so, but in terms of evolution, so if we have look at back a long time scale, so how this symbiotic relationship formed, it's a very interesting question. So the second is so how the mechanism, how the co-microbiome maintained, as César just asked me. So I think this is another open question. So the third, I think that, so the integrated biological, biogeochemical processes that the living carbon drive microbiome mediating soil nitrogen and the phosphorus mobilization and the turnover, and their agricultural, ecological, and environmental consequence. So that is a very also very interesting questions that need to be conducted. Lastly, I think the new method innovation. So the research on hyphosphere interaction network will encompass the environment, plant, and AMF fungi and bacteria. So this need to be achieved by upscaling AMF fungal bacteria interactions from lab to the field. Therefore, it's very important to create some new method in the future to make the hyphosphere effect and the process visible, touchable, I mean, and real-time measurable. I think that's such kind of new method will be very necessary. So for my personal interest, so I'm currently working on the signals that comes from fungi that induce the bacteria moving toward the fungi and along the fungi hyphae network to some soil patches, like organic patches, inorganic phosphorus patches. That's we are currently working on. So hopefully we will get some new stories in future. Right.
Wow, that's quite amazing, and I am very happy with this interview. It was a lot to get into the mind, but it was so exciting. And thank you, both of you, for your time, and thank you all.
Thank
Bye.
you. Very, very
And
interesting.
thank you. Thank you. Yeah, thank you for your good question that make me think deeply, thinking further. So thank you again.